What do you know about dementia and its relation to iron metabolism? Let me help you with that. The two types of iron dyshomeostasis are iron deficiency anemia and hemochromatosis. Knowing this, a clear link will be formed to the need for future research on the relationship of this topic. Key elements include iron and its importance for human health, dementia stats and epidemiology, and the pathology and neurobiological mechanics of dementia. What is the importance of iron? Iron is the most profuse metal in the body, being essential for blood production, oxygen transportation, mitochondrial respiration, and neurotransmitter synthesis. Efficient cognitive function results when iron homeostasis in the brain is achieved due to optimal functioning of these mechanisms. Too little or too much consumption can be detrimental to our health, leading to iron dyshomeostasis, neurotoxicity, and oxidative stress through the destruction of neuronal cells, causing conditions such as dementia. Now, time for some figures. Globally, every 3 seconds someone develops a form of dementia. In Australia, 500,000 people suffer from a subset of dementia, being the second leading cause of mortality and third leading cause of disability. Holding an annual $60 million financial burden, continued research is required to address the expected growing incidence rate of dementia due to the high rate of morbidity in the aging population. What is dementia, and what are the related subsets and relevant neurobiological and pathological mechanics? Dementia has relation to nerve damage in the brain, resulting in cognitive decline, symptomized by memory loss, personality changes, and reduced concentration, occurring mainly in the elderly. As we age, brain cognition and vasculature decreases. Consequently, the permeability of the blood-brain barrier increases, resulting in greater accumulation of ions, molecules, and cells in the brain. The blood-brain barrier's role is to protect the central nervous system from pathogens and toxins, as well as regulate movement to avoid excessive accumulation. This function results in the onset of these disorders. The main subset of dementia is Alzheimer's, with vascular, Lewy bodies, and frontal temporal dementia being the following most common. Each of these subsets contain different causes, but derive from the same process of neuronal cell damage. How do these elements relate to our understanding on iron metabolism with dementia? Iron deficiency anemia is known to develop dementia. It is characterized by the lack of oxygenated blood supply to the brain, resulting in neuronal death and tissue damage. This has strong links to hemoglobin function, being the transfer of oxygen around the body, as each heme consists of 4 oxygen molecules. Related studies link iron to blood production, as 70% of our body's iron is stored in hemoglobin. Essentially, with poor iron metabolism, low hemoglobin efficiency occurs, causing insufficient oxygen and blood reaching the brain, resulting in neuronal damage, subsequently being the grounding cause of vascular dementia. Similarly, previously completed studies by Hyung Hong and John Saint both conclude that hypoxia has a strong correlation with increased cognitive decline, stating the necessity for further research into anemia and dementia. Contrastingly, hemochromatosis is excessive iron accumulation. This notion is specifically linked to the blood-brain barrier process. With additional elements of tau and amyloid beta proteins, the catalytic Fenton reaction can be formed to link this topic. It is known that iron is transferred across the blood-brain barrier into the brain precisely in specialized tight junctions found within the endothelial cells through special proteins called divalent metal transporter 1 and transferrin receptors. Transferrin then binds to ferrin, a protein that specifically stores iron. Essentially, cognitive decline occurs due to the increased brain toxicity and oxidative stress through lower functionality of these factors as we age. Tau and amyloid beta are related proteins that cause dementia. Amyloid plaques and neurofibrillary tangles develop in the brain through the binding with iron and passing through a permeable blood-brain barrier. Studies have found that plaques disrupt the integrity of neuronal processes, whilst tangles disrupt protein homeostasis, but this is still debated. Overall, greater iron production causes greater tau and amyloid binding, resulting in this toxic accumulation. Studies believe that tau hyperphosphorylation and plaques occur due to oxidative stress. A study conducted by Rao has recognized that greater studies are required to support the link of tau and iron, whilst Kamatani has stated amyloid plaques are not relevant to iron dyshomeostasis and Alzheimer's. But where does the Fenton reaction come into this? The Fenton reaction is recognized as the phenomenon of hydroxyl radical generation due to abnormal iron metabolism. Contrastingly, this reaction triggers oxidative stress, causing cell damage. Similarly, iron acts as a catalyst to this oxidative process. Essentially, the more iron there is, more Fenton reactions occur. The more reactions that occur results in more cell damage due to inhibition of these free-forming radicals. This presentation has consistently affirmed and related to the need for future research into the topic. An abundance of literature regarding the two covered conditions are present, but studies lack highlighting the relationship between mentioned factors. Furthermore, presented literature is contradictory. Conflicting perspectives from Uranga and Kamatani show that amyloid beta and iron binding can and cannot be relevant to Alzheimer's development, thus asserting the inherent need for further studies. Hoskins supports this, as she calls for further studies to provide a more wholesome understanding. Without this, necessary development in our understanding and support towards the prevalence and financial burdens of dementia will not advance. To progress, further proposals should be implemented to fill gaps in literature. Thanks for listening.